you for confirmation. Some issues related with our Zoom server. Maybe you heard that at Makandura, even our computer unit has been inundated yesterday. Hope you can hear me still.
Yes, sir.
Okay, right. Thank you. And sorry about that delay. Maybe you heard that even that your final year's repeat presentation scheduled to be held today has been postponed due to these bad weather conditions for both students who are presenting today and for the organization at Makandura, there are issues. Right. So anyway, I'll now once again share the presentation and we will continue our uh, meeting. We'll try to continue. I hope there will be no issue now. Right, as you can see, in addition to what we have discussed uh, last day, that is regarding the insect pest management. If I just summarize what we have discussed that day, I'm not going to repeat. Don't worry about that. We were talking about insects, their biology and ecology, because uh, before starting the uh, discussions on the management of pest, it is necessary to understand how that problem is developed, the pest problem. And then after discussing that, we understood that the pests means the consumers of plants in nature, and those consumers are increasing their population, increasing their consumption of our plants, and because of their increased consumption, they cause economic losses to our crop production. So because of that, we name them as pest, or the organisms interfering with us or competing with us for the consumption of our crops. So in natural environments, the population of those organisms, primary consumers, or now they become pests, they are controlled. The populations are controlled by natural factors. So by studying their biology and ecology, we can understand the factors controlling their populations in natural environments. So that is why we are studying about the biology and ecology of those organisms who can become pests because of the necessity of managing their population. So, in order to complete that task, we studied about insect biology and ecology. That was one of the major groups of pests. Then we know that there are other groups of pests. Some of them are having more familiarity to us. So about the insects, we had no studies in our course earlier in our degree program. But about the microorganisms were studied by you earlier in the microbiology subject. Maybe you had some other studies about the microorganisms earlier as well. So in our program or in diploma programs, you have followed in our faculty or diploma programs, you have followed in a different place, or maybe at school in the biology subject, there were some studies on microorganisms. But now we are studying about the microorganisms who are causing plant diseases. We need to study about them. So, because of that, I have selected this topic for today's session as one of the main topics for today. So, that is about the plant pathology. So, the topic is there, plant pathology, the fundamentals, the important major concepts of plant pathology. So, what is the meaning of plant pathology? Pathology in Latin, so the Latin language is the basis for many scientific terms. So, plant pathology means pathology is the studies on diseases. Pathology. So, plant pathology means study on plant diseases. I know it is clear for you. So, as it is the normal way of starting a discussion on topic, I have described the topic, the meaning of the words, terms we are using, and then So now we are to study the diseases of plants. So all of us had the opportunity, fortunately or unfortunately, we all had the opportunities to study the diseases of plants, or at least observe to see the plants that are with diseases. So I think in your visits to your own, or where you are working, the tea plantations, or as a visit to maybe the areas of tea cultivation of country, you had, or you may had the opportunities to observe tea plants. If you are getting closer to the tea plants, maybe you had the opportunity to observe normal tea plants like this, and the plants with some different, changed, or abnormal appearance on the leaves or other parts of the tea plants. So we have not mentioned the examples of tea plants with the insect pest damages, but I hope you study about the tea pest of tea plants during your studies in tea agronomy. And similarly, about the diseases of tea plants also we had studied earlier, or at least observed, right? So our topic, fundamentals of plant pathology, the study on diseases, and I am mentioning about the diseases of plants that are to be controlled or managed by us. So what do you mean by a plant disease? Now, this appearance, I think, is indicating us that this plant is having a disease condition. Then here we can see only the leaves of this plant, but sometimes not only a leaf, maybe the whole plant can have some abnormal appearance like this. So about our understanding of a plant disease, what we consider as a plant disease, why we consider some of the plants are having a disease, or some of the plants are healthy plants. So according to your observations, the A and B, two plants are here. Now, for the first instance, or at a first glance on these two plants, which plant we consider as a plant with a disease? Your opinion, please?
B.
Of course, yes. Plant B is, it looks like a diseased or diseased plant, or plant with a disease, right? So without stopping at that, identifying it as a plant with a disease, so with that answer, now shall we justify our answer? Why we consider the plant B as a plant with a disease, that there is a disease? How you consider it as a plant with a disease? Can you please give more elaboration, more justification? Why plant B is having a disease. Why you say like that? Not only the student who mentioned it as a plant with a disease, all of you can join. Why we consider plant B as a plant with a disease?
Looking like unhealthy.
Looking like unhealthy. So why we decide that it looks like unhealthy plant?
We can see curved leaves in plant B.
Leaves are not normal. They are curly. Yes, Roshan, you want to say something?
Lack of plant nutrition, the leaves are scrolling.
Right. Okay. You are telling about reasons for rolling. So it can be due to some nutrient issue. So one of the reason we consider to say that this plant is a plant with a disease is the curviness, rolling of the leaves or abnormal appearance of the leaf or the leaves, right? Rather than leaf, it is all the leaves are having some unusual appearance of rolling, curling, right? Then why the change of leaf shape can be considered as a reason to say that it is a plant with disease. What is the trouble for the plant? What is the difference for the plant if there is a change of the leaf shape, appearance?
So water deficiency reflection.
Now again, Roshan, now it is good that you are mentioning several reasons for the change of the leaf shape. That again, yes, lack of water or the deficiency of water, drought conditions may change the leaf shape, the leaf vigor. That is true. So the change, my idea is to understand why uh, change of this leaf shape is considered as a disease condition. The reasons you mentioned are perfectly correct. Lack of some nutrients, lack of water. Now, if we compare now in the leaf, in this plant A, leaves are, we assume that it is having normal appearance compared to the plant B. So that is why you all are mentioning there is a problem here, disease here, compared to this plant. So Roshan or those who mention about the opinion that B is the disease plant, I can't remember your number, but can you say why the change of leaf pattern, leaf shape is telling us that there is a disease in this plant. What are the changes? Now, the change of the leaf appearance is something we observe from outside. Internally, what are the differences in these plants? Maybe due to this change of the leaf shape, appearance.
So pesto overloaded by nutrition.
So this is a stress condition. When leaves are having this type of different shape. So due to this different shape, if you compare these two, now externally one difference we have already highlighted, and even we are telling the reasons for this difference in the leaf. Now, due to that difference in the leaf in these two plants, any internal things you can mention?
Damage cells in the plant.
So they are, due to some damages to the cells or tissues, the leaves are changing. Yes, so that is something internally going on. What about the functions of the plants?
It is fungal disease.
This can be a fungal disease, but now what are the things? So it can be a fungal, it can be caused by some other organism, but the internally, what those maybe a fungus or some other type of organism doing changes as internal, especially the functions. So now suppose that maybe you can hear a change of my voice. Now actually the last day it was worst. Today is a little bit more how can you say, I'm in a more healthy condition. Sorry about lack of the video. Anyway, I know that video is not necessary, but so I am telling, I have some changes in my voice, and last day I had the feel of high temperature when I was discussing with you last Saturday. So and maybe running nose also I had. Now those are the things we can observe when we are having a disease, but internally we have some additional things, some different types of functioning going on when we are having a disease in our body. Similarly, in the plants, now we mention here that there are leaf diseases in the plant. Here, there are no leaf diseases or disease symptom in this plant. So, you are mentioning several correct reasons for possible reasons for these changes. Now, good that we are clearly understanding the reasons for this change. But what are the things really happening here and happening in this plant as well? What are the differences due to this changes of the leaf appearance. Right. Okay, please.
Plant can't maintain normal mechanism like photosynthesis and can't maintain growth rate.
Oh, yes. So, leaf or the leaves are the photosynthetic apparatus, perfectly correct. And if the leaves are having a different shape, appearance, structure, that means photosynthesis is affected. It is a major function in the plant. And if the photosynthesis is affected, the production of dry matter, synthesis of materials required for many things, mainly the required for respiration. And in addition to that, when the fruits or other parts are produced by the plant, it will be affected due to the reduced production in the photosynthesis, and it may be creating reduction of the yield, and also the as a reaction to the attack by these whatever the pathogen, maybe a fungus, maybe a virus, maybe a bacteria. Like in our body, plant is increasing the respiration rate as a reaction, and there are many reactions by the plant for this infection. So the respiration is changed inside the plant. As you mentioned, in this plant, there are unusual, abnormal deviation from the normal functioning, like functions of respiration, especially the photosynthesis, is changed in this plant. So, a plant disease means the deviation from normal functioning by the plant. And it is common for other organisms, including us. And as you said, maybe lack of water, lack of nutrient, or other factor, change of the availability of those factors will be changing the normal functioning of the plant. If it is water, then lack of water creates some changes, maybe like this, so little bit different than this, but the leaves are getting droopy. We call the wilting appearance or the dehydrated appearance. So those changes in the leaf is many changes due to lack of water, including this type of appearance. So, but if we can rectify that condition, or if we can supply water without a delay, because if it is too late, maybe plant will completely die. Otherwise, if it is quickly possible to supply water, or if water is given by natural supply of water through maybe rains, then plant become normal again. That means the deviation from the normal functioning due to water can be corrected, rectified by giving water. So it is a temporary deviation, but sometimes these changes are appearing, and even if we give water or any other input, maybe it may not come back to normal conditions. So the continuous deviation from normal functioning is the condition that considered as a disease. So actually I thought of asking or discussing about the reasons for the plant diseases or causes of the plant diseases, but you have mentioned them correctly. I only would like to group them according to their liveness or their biotic or abiotic condition. So the causes or the reasons for plant diseases, as you mentioned, it can be water, light, some toxic chemicals, nutrient deficiencies, pH of the soil or other medium, change of temperature, and there can be living factors like fungi, bacteria, viruses, viroids, phytoplasma, parasitic plants. I assume that you had some studies on these biotic factors as well as these abiotic factors. So when the disease conditions are caused by abiotic factors or non-infectious factors, that type of disease, for an example, if these conditions are caused by lack of water, as you suggested, then the condition will be available with the plants that are not having water at sufficient level. But that problem of lack of water will not be given from one plant to another plant. It can be available with other plants if they don't have water. So these factors, causes, if they cause a disease, those diseases are not transmitted and we name them as non-infectious diseases. But if it is caused by a living organism, that living organism can move into another plant, another healthy plant. So because of that, we name such a disease as an infectious disease. So infectious disease means a disease that is possible to be transmitted from an infected plant or from a plant with the disease. So when an organism is causing a plant disease, we name such an organism as a pathogen, generating a condition, pathological condition, disease condition. So an example is appearing on the screen. Similar type of diseases may be not only on tomato, on other crops. You have produced, grown at your organization, in your farm, in your home garden. You may have observed several large number of plant diseases. So you observe the diseases like this. So this can be caused by different types of organisms, and I hope you have observed and studied about the disease conditions caused by these abiotic factors, because you studied about plant nutrition, soil management, plant nutrient management, and as we mentioned earlier means during the last day, about our consideration of biotic factors during the subject crop protection. So we are considering biotic factors of plant diseases to be controlled by us, because we know that as a good farmer, good planter, good agronomist, we are managing these factors in a way that they are not causing any trouble. If we are not managing well with the water level, temperature level, light level, or all these factors, then they can cause some non-infectious disease conditions. So managing those non-infectious disease conditions are being discussed in other subjects, so we are not considering about them here. But about the pathogens who are causing plant diseases, who are generating a plant disease, has to be considered by us in detail. Why? We want to control the plant diseases, otherwise the plants will be having abnormal functioning, deviation from normal functions, and that will be reducing the productivity, yield of our crop plants. Right. So now, as we studied about the biology and ecology of the insects, that means we need to study about the biology and ecology of plant pathogens also. Now, we can study the biology of fungi separately, biology of bacteria, biology of viruses. But we want to know the aspect of their biology that create plant diseases, isn't it? Because if they live normally without creating a plant disease, that is okay. But when they are causing a plant disease, then only we want to manage them, because we are unable to 100% remove the organisms that are causing plant diseases from our environment or from our agroeco system, production system. But we would like to keep those microorganisms under control so that they are not developing a plant disease in our crop. So, to control the disease conditions in plants caused by microorganisms, or to prevent the disease conditions like this in our plants, we need to understand how a pathogen is developing a plant disease, how diseases are generated inside a plant. So, that process is necessary to be understood by us if we want to manage the plant diseases correctly. So I would like to think about that process. I would like to discuss with you about that process of developing a plant disease, because we want to prevent the development of plant diseases in our crops. So that is our next topic for the day. So there are different types of plant diseases. Diseases on the fruits like these, diseases on the stem parts, branches, or maybe diseases in the roots, and diseases on the leaves. I hope at least you heard about a disease called late blight in potato, Paschima Angamaraya or Angamara Rogaya. So, if you need to understand how to control these diseases, that means we need to understand how these diseases are controlled, how these diseases are developed, because controlled disease condition can be achieved only if we understand how the disease has been developed. What are the reasons for the development of a disease? So that process we are going to discuss now. I hope you will actively participating or helping me to develop the discussion. So when we are discussing, then you will definitely feel that you know what is happening. Only thing is, we have not considered these processes earlier, or we have not considered the necessity of studying or understanding that process. Maybe you are well ahead about the understanding about this process, right? So, if we just consider the process in steps, one thing I would like to highlight, even though we are talking about the steps of the process, like given here, 4 steps are given. Inoculation, first step. Second step, pre-penetration. Third one, penetration. Fourth one is the infection. Then there are other steps. So at the infection, it can be at a higher level of activity, but then there is a step called growth and reproduction of the pathogen, dissemination of the pathogen, and after disseminating, the pathogen is surviving in the environment. So what I was trying to highlight is that even though we have mentioned the process in 7 steps, 7 stages, these are actually going on as a cycle. These are cyclic events. That means starting from first one, process is going on, second, third, fourth, fifth, sixth, and seventh step. But as it is a cycle, after this 7th one, the first one is coming quickly. Or once we discuss about this process, maybe one of you can suggest, or many of you will think that why don't we start the discussing this disease development cycle from the step survival? Why not? Yes, we can start the discussion on plant disease development, or we can study the disease development from the place of survival. That is true. So I thought of studying with the step inoculation. So even when we are talking about the inoculation, we have to refer to the place of survival as well, right? So in these photographs, you can see the diseases on the plants or on the harvested fruits, or maybe still the fruit is attached to the plant as well. Or maybe you have seen the diseases on these banana leaves, and the banana grower would like to control this. So maybe he will try to prevent the disease if possible, or if it could not be prevented, the appearing disease could be controlled if the person growing this banana is understanding the disease development, how it has been developed. And another thing is, studying this process will tell us, when we are observing a disease, we can understand that what is the stage of this disease according to the disease development process. Of course, on our plant, on our crop, maybe we are observing diseases at all stages, including the survival, because some of them are surviving on our crop plant as well. Right, okay. Then, when we are discussing about the plant diseases, disease development process, there is a important thing to be considered when considering any organism that is having a contact with our plant, maybe an insect, maybe an animal, ant or elephant or microorganism. It is having contact with our plant and causing a trouble. So when we consider a plant disease, it is a trouble caused by three components. Maybe when I am telling like that, you can argue how the host plant, how the plant itself can cause trouble to the same plant. Are the plants causing troubles by its own? Is it possible? Right? So it seems like that we are now, according to that idea, we, human being, myself, so I myself is developing a disease in my body, or a plant itself is developing a disease in the plant. Is it the meaning? Actually not that plant or a human being like me developing a disease intentionally, or we would like to develop diseases in our body. No, but disease development is a process with the participation of all these three components, right? So we usually say that the pathogen is causing a disease, but where it is causing the disease? In the host. Then where the host is living? In an environment, in an ecosystem. where the pathogen lives in the same environment or in an ecosystem. If they are living in two different ecosystems, then there will be no disease developed by pathogen in the host because they are in two different places, right? So they are to be in the same environment, same ecosystem. Then more important thing is that if they are living in the same environment, that means the conditions of the environment are having influence on the development of pathogen, and because of their development, it can cause the trouble to the host. So the vigor of the pathogen, vigor of the host, the strength to fight with the pathogen by the host, and strength to the attack needed, strength to attack the host needed by the pathogen is also decided by the environment. That means a plant disease is a condition, a phenomenon that is equally disease is a condition that is equally developed or equally influenced by these three components, right? So we will come into this illustration again once we discuss the disease development process, right? So if we go through the process, starting from the step I mentioned as the first one, maybe later, once we complete this, you will explain this to some other person by starting with the step survival place of the pathogen, but I prefer to start with the inoculation step. So it is not that inoculation is the very, very first step. Maybe in our agroecosystem, pathogen is surviving somewhere, maybe in the plant itself, but we are not observing it. Nothing is observed, visible. So inoculation, if we consider it as the first one, so what we understand by the term or step as inoculation. So suppose that my plant part, plant product is not having any problem, any trouble related with a disease. But as we mentioned earlier, it can be caused by a pathogen. So if where my plant, the host of the disease, is living in that environment, if there are pathogens in that environment, but if they are in two different places, there cannot be a disease, isn't it? There are not no remote controlled disease development, pathogen has to contact the host. So, the contact, that simple action, which is very important and necessary for the disease development, is considered as the inoculation. So, what is inoculation? Contact of the host by the pathogen. And that contacting host part or host stage should be active, live, or viable. Otherwise, it will not be capable of developing a disease, right? So, because of that, we better to say inoculation means contact of the host plant by a viable propagule, viable stage of a pathogen. So, type of diseases depend on what type of pathogen are contacting our host. And there is another thing. Now, we mentioned that there are hosts, there are pathogens, and they are interacting in an environment. So, the interaction is creating a disease. So, the first ever interaction, the first ever plant that is developing a disease is we are observing maybe first. So we mentioned that inoculation means contact of the host plant by a viable, live, vigorous stage or part of the pathogen. So that viability means the possibility, capability of attacking the plant. So that type of viable or live pathogen stage is considered as inoculum. What is inoculum? The viable live part of the pathogen that is capable to start the disease development process, that is completing the inoculation. It is named as inoculum. So the inoculum that initiate the disease first is considered as primary inoculum. It starts the primary infection. But once the disease or infection has been started in a part of the plant or in a plant, the very first step is the primary inoculation. Primary inoculum is causing primary inoculation, that is causing the primary infection. But from that primarily inoculated plant or plant part, there can be secondary infections or secondary inoculation happening after the primary one. So if we explain it a little bit in a different way using some examples. So now this is a disease condition in a harvested part. You all had observed this. Actually, the name of this disease, anthracnose. The symptoms are like this. So, out of these fruits of this bunch, maybe one of this fruit started disease development first. So, from somewhere, this pathogen that is causing this disease has arrived, right? So, the first ever pathogen stage that is coming into the contact with the host plant is considered as primary inoculum, that is causing primary infection, right? So, when you say primary infection, sometimes in some books it is explained like this. So, the infection that is started on a plant part very first, primary infection. Then, so this infection is going on, and maybe from this fruit, it is transmitted to some other fruits that are in the nearby place. So, on other fruits also, this black color patches are developing. So, initially it develops in a different way. So, it is not that we are explaining the different types of diseases. Now we are explaining the disease development process. So, this, assuming that this one is with the primary infection caused by primary inoculum, and from this plant, this plantain, this fruit, it has disseminated, sent the pathogen to other fruits. So, the infections started after the first one is considered as secondary infection. But there is another type of secondary infections. Sometimes when the primary infection started like this, so this is the disease started first, the brown color patches when anthracnose is developing, when these patches are joining together, we call amalgamation is going on. The whole fruit is covered with that discoloration, changing into brown. So, so it spread. And in the places where the diseases started first, the plant tissues are deteriorated, getting dead condition or death tissues, cells. We name them as necrosis, necrotic tissues. So, on the dead cells, dead tissues of the plant, maybe a secondary attack by a different pathogen can happen. That type of an infection is also named as secondary infection, right? So, those two types of primary and secondary infections, I'll explain using another example. Now, this is a situation with plants, not a fruit. So, earlier we discussed about the fruits from this plant, right? So, you can see certain higher number of banana plants are appearing here. And on the banana leaves, there are leaves with a disease symptom. So we can see a disease here on these leaves, here also, like that, right? So, just to explain, I assume that, I think you also agree that this plant leaves are having higher level of the damage. So, these leaves are covered with the disease at higher percentage. Here also is the same, but in this plant, on this leaf, the infection or the damage or the disease is appearing at this tip or end part of the leaf, right? So, I assume that this plant has been infected, inoculated first. So, from a place of survival of the pathogen has arrived here somehow and started infection. So this plant has been infected first, and then when the disease is developing in this plant on these leaves, the pathogenic organism, this is caused by a fungus. So this fungus is developing here, and the propagules of this fungus is transmitted to other plants, leaves of other plants. So the viable propagule contacted this plant first, was the primary inoculum that started primary infection in this plant. Then from this plant, pathogen propagules are moving into other plants. So the propagules of the pathogen released from these leaves of this plant, they are secondary inoculums that are contacting these neighboring host plants. And those secondary inoculums are starting secondary infections on some other host plants. Maybe if we assume that this is the first plant infected, so primary inoculum produced by this plant has been disseminated into all these plants, and now on these plants, secondary infection is going on, caused by the secondary inoculum, right? It is very important to understand this, why we are studying. I am repeating what I have mentioned earlier. We are studying this process in order to control plant diseases, protect the banana plants from these types of diseases. So, we need to prevent inoculation. We need to prevent these types of banana leaf diseases. So, if we want to prevent, if we want to control, stop the diseases, we need to understand how those processes are going on. So now we understand the primary inoculum that cause primary infection, secondary inoculum that cause secondary infection, right? So another example, but very quickly, a tea bush looks like a healthy plucking table, tea bush with healthy leaves. Then if some other plant is having this disease, can anybody name this disease? So I didn't ask about the banana disease. Maybe you know the disease appeared here. Can you name this quickly? This disease? it is banana sigatoka disease. Then this one on tea leaves, maybe we have some representatives from tea industry. This is blister blight disease. So somewhere in this plantation, there are few leaves in this plant having a disease. So how do you think in this plantation, if this is the first plant having this disease, it is primary infection here. So during the development, if this plant with the disease is having this symptom, this disease, and the pathogenic organism, if it is going to contact a healthy plant with some viable propagule part of the plant pathogen, that viable part that is possible to start infection in these healthy plants, it is mentioned as secondary inoculum. So a primary inoculum has started disease here, and from primary infection in this plant, secondary inoculum has been sent or disseminated to new host plants, and the secondary infection will start here, right? So we mentioned that there is an inoculation going on. To start the inoculation, inoculum has to contact the host plant. So from where those inoculums are coming, what is the source of the inoculum? We need to know that. Why? If we want to prevent the inoculation, if we want to block the entry of inoculum to our cropping system, we need to know from where the inoculum is coming. So the place of inoculum storage, survival, it is named as source of inoculum, right? So actually, these are the places of survival of the pathogen also. So we mentioned about the survival stage as the last step of the disease development process. So potato tubers that are infected with the pathogen, right? So we usually mention that the potato leaves are affected by the late blight pathogen, but this pathogen can grow inside the tubers also. So from these leaves, the pathogen can go into healthy potato plants. Then these tubers can be keeping pathogen. So from these tubers, if a new plant is grown by us using a tuber with the pathogen, so that pathogen will automatically get into the new plant also, right? So the leaves with this disease, when it is fallen on the ground, or the plant parts with the pathogen, they are plant debris with inoculum, so a source of inoculum. Tubers, they are propagative organs. as well as food also. But infected seed tubers of potato can be a source of inoculum. Then the instruments we are using when we do ploughing, maybe manually or using an instrument, maybe we are contacting the fallen leaves with the fungus, and that if we use the same equipment, maybe a bucket that contain the tubers, a tray, a bag, it can be a source of inoculum. Then we know that some viral diseases on plants are appearing like this. Then not only the crop plant, some wheat plants can also have the same disease. Then those weeds can become source of inoculum. Then the surfaces of the infected plants like this. Maybe the plants are living with the disease, so the diseases on these leaves developed by a pathogen. So from the surface of this leaf, an inoculum can go into a healthy leaf of the same plant or healthy leaves of a different plant. Right. So I was talking maybe a little bit too long. So I mentioned about the sources of inoculum, and I mentioned that the inoculum in these sources are contacting the host plants. That means they are going to the host plant. How they are arriving? Because we know that they are microorganisms. Can they move independently, or do they need some help? How the pathogens in this, if this is a wheat plant, how it can go into a host crop plant? If this is an infected potato plant, how the pathogen can go to a healthy new plant? How they are moving, how they are landing, how they are arriving on a new host plant. Can you help me to list the method of their arrival? Please. I know that you all know very well about how the inoculum can go into a new host plant. There are methods of movement. Please do not hesitate.
From insects.
Yes, of course. There can be some insects who are, especially when the infection is with the virus, when the insects are feeding on the infected plants, they are taking the plant cell sap, juice, or whatever the plant part, and they will be contacting the insect, maybe their legs, maybe their mouth parts. Now, I think if you read the section on insects in our module, so we are mentioning about some insects who are uh, sucking cell sap of the plants, and those sap-sucking insects are, of course, taking the propagule viable part of the pathogen to a healthy plant, just like mosquitoes transmitting the diseases. Now, these days, we are talking about dengue fever. So, yes, perfectly correct, and maybe one of the most important mode of landing inoculum you have mentioned. Right. Okay. Other methods?
From natural source, wind, water.
Yes, natural factors like wind, then water. From how do you think? Water, there are many natural sources, so ways of receival of water. Even we are applying water to the plants. So what type of water you mentioned? It is perfectly correct. So while you are thinking about that, if I'm telling, so when the wind is blowing through the canopy of these plants, the spores of this fungus can be taken with the wind and placed on a plant somewhere else, or even on a leaf of the same plant or leaf of another plant through the wind. But what about water? Because water in this plant will not go into other plant. I think like that. But how do you think that water can take the pathogen into a new place? I feel that it is possible, but if you can give more details. Right, okay. Anyway, it's good that you have mentioned the major methods of landing, arrival of inoculum through the wind, and then vectors, these types of insects who are sucking sap from this place. And when they are moving to another plant or another leaf of the same plant, they can transmit. And then now regarding the water, now when we do irrigation with the overhead irrigation method like sprinklers, or any method that create water drops, or maybe when the rain drops are fallen on a place like this with a disease. So due to the gravitational force and the speed of the drop, raindrop or irrigation water drop can be broken into small droplets, right? So when the drop is fallen on this place, instantly the propagules of the pathogen is contaminating that water drop, and the broken droplets, they are fallen here and there. So the water droplets are with spores or the inoculum of the pathogen. So it may start new infection. So from the primary infected leaf part, there can be a secondary inoculum going into a new place. Right. So we have mentioned about the first step only. Why we are talking about the disease development process? We are talking about this process to control the plant diseases, to manage the plant diseases. So, how to manage this process? Now, as we have studied about, or we have discussed, maybe very briefly, we have already mentioned about the first step. So why don't we think about controlling, managing the first step? How to prevent the first step? So we were talking about how the inoculum is coming, the mode of landing. We were mentioning how from what type of places inoculum is coming. We were mentioning how the inoculum is developing a new disease starting. So inoculum is the starting point, contact of the host by a viable step of the pathogen, right? So we were talking about sources of inoculum, everything about the inoculation, right? So while giving a idea to think, I am giving a break as well. So I request you to have a break with some thoughts of possible controlling of the inoculation. So it is the only starting point. Maybe one can say there is no disease to control, but why don't we think about controlling now or preventing the further steps? Because in our health sector also there is a saying called the prevention is better than cure. So in plant diseases also the same. So we can now think about mentioning, explaining how to control plant disease at the step of inoculation. So your ideas will be very valuable, very helpful. So after the break, we will start with those thoughts. So please think about these possibilities of prevention of the inoculation during this break. So we'll have a break for 15 minutes. but it is better to mention how the inoculation step can be avoided. How to control the inoculum? Is it possible to have some control on inoculation? So please, your suggestions, let us have some time allocated to that, because it will be easy for us to do like that, rather than discussing the management of diseases at the end, because now we have the fresh memory on inoculation. I think you can mention a lot of methods.
Use clean planting materials.
Right. Okay. Now with the planting material, how you are correct. First of all, I have to mention it. Then how to what type of diseases? So why you think that clean planting material will be preventing inoculation? Can the planting material of crop with some contact by the inoculum or contaminated already? So why not? Now, potato tubers that are produced by infected plants may contain the pathogen. So, so don't be, I mean, feel shy or may no confident about your answers. So you can justify always what you think as correct. So the tubers produced by an infected plant like this may contain the pathogen or with the symptoms developed like this. So not only that, the plants with viral infections, they produce seeds or vegetative propagules with the virus. So because of that, it is necessary to get materials without any contamination, contact with the inoculums. So that is, of course, very valid and efficient way of preventing inoculation already. So with the beginning of the crop, there is a possibility of having the infection. So please think there is a, message from our office. I'll check it, please. Have you thought about other methods in addition to the clean planting material? Please add something more, not just something. Other methods of preventing inoculation. There can be a lot of methods. Right, okay. If you take more time, then oh, please, please. Please, number 141. Right, okay. Then if you are not taking that opportunity, so I'll also not spend much time on that. But I'll mention just one and let you do it independently later on. So the why don't you think about the possibility of removing these type of sources? If you know what are the sources, if the sources are the place from where inoculum is coming, if we can have a cropping system without these sources, it is a possible way of preventing inoculation. And if we know the methods of arrival, landing, why don't we block these methods? So controlling vectors, insects is one way of preventing inoculation, right? So anyway, I think it is maybe, so with my last week experience, I could have thought that you are a bit reluctant to use the opportunity of joining. or maybe you think that no need to help me. Actually, it is helping you more than me. So it builds your confidence. It builds your logical thinking. For me, actually, it is maybe easier to go and quickly finish. Maybe you also like that. So anyway, I have another suggestion or advice. So when you read the disease management methods in this module, or maybe uh, in the modules where you study the crop agronomy, crop production subjects in our course, you can see a lot of methods of plant disease management. For an example, when you study tea agronomy, there will be some details on insect pest of tea, diseases of tea plantation, and there will be a list of methods of controlling, protecting from those diseases. So you go through those methods and check what type of stages of infection process is targeted by certain action. For an example, as you mentioned, in a crop, as a method of controlling a disease, there can be a method recommended as use of clean planting material. Now, we are learning this subject to understand how clean material, clean planting material can be useful for management of a plant disease. So I explained it very briefly, but you can read and think more in detail about how to connect, link the different methods of diseases and pest management given in different modules, in different literature sources, in crop production guidelines. Then you will be able to suggest by your own the methods of pest and disease management. So, okay, I was trying to get some suggestions, ideas from you, but only a few listening. Maybe that is also possible, even though a small number of your group is connected today. Now it is 30, around 30. So out of 30, maybe only a few students are actively participating. Others are just maybe connected. So, right, then the second step, once the inoculation completed, that means the pathogen has already contacted the host. Suppose that this insect has brought a virus into this plant. Maybe the train droplets have disseminated spores of this pathogen fungus onto another plant surface of this type of a plant, right? Now, the pathogen is there on the contact with the host. So, host is there, pathogen is there, but there is no disease yet. So, there can be some possibility of developing a disease just because of the inoculation, we can't say that there is a disease already started. But no need to take a risk of having inoculation, because with the inoculation, there is a possibility of developing a disease. How it happens? Still, the pathogen is there with contacted host. Whether the disease is developed or not, it is not sure yet, right? So, in order to develop the disease, the pathogen has to have a very good contact with the host. And if the disease is to be developed inside the host, the pathogen has to enter into the host. So, that is the next step. If we say enter, it may be entering through to an opening, a door. If I say that I am going to enter into an auditorium, that means I am entering through a door. Then sometimes we say that some thieves have stolen some goods, valuables from a house. Then usually the thieves, they are not entering as we enter, because maybe they are entering, the cunning fellows, by cheating us, they will enter through the open doors, and after entering, they will do their stealing the valuables or whatever the thing they want. But most of the time, either they are getting through the broken doors, windows, or they are entering by force. So in that case, we use the term or the word intruders, or some forceful penetration. So the next step, as we mentioned in our list of the stages of disease development process, it is the pre-penetration step. So the penetration we consider as a kind of entering with the use of some energy, force, power, not just entering passively, right? Some pathogens, of course, they are entering through open gates or openings of the plant. That is true. In that case, we can mention as entering. So now the spores of a fungus, like this. Those spores are produced by fungi. Actually, the leaf diseases or anthrax node diseases caused by a fungus called Coletotrichum, and they have the structures like this to produce spores. So they are spores. Whatever the fungus, when they contact the host, they will be on a surface of a plant organ. with the spores of fungi or with the propagules of fungi, the entering process or penetration process is more complex. And compared to other groups of plant pathogens like bacteria, viruses, the pathogens are having more complex disease development process. So because of that, I am taking the disease development process of fungi as an example, because the disease development by other groups of pathogens are bit simple, with less steps and clearly understood compared to the fungal disease development process. Therefore, we will discuss about fungi, their disease development, but I hope you will read the disease development and disease management in relation to other pathogen groups as well in our module also. Right. So, when a spore of a fungus located on a plant part, especially a plant leaf, it looks like this. So the spore appearing on a plant surface like this. So this is a real picture. This is a drawing. So the spore is still on the surface. Plant cells are here. If it is a leaf, it is covered by a cuticle layer, and spore and the host cells and tissues are now in contact, but nothing happens yet. The spore of a fungus can cause a disease only if it is having some kind of interaction with the plant cells tissues. How it can interact if it is still outside? From outside also there can be some interactions, but no change of physiological, biological processes of the host. So, spore is starting now developing itself. Just like a plant seed absorbing water, it start activation of enzymes. For that, they need optimum temperature, humidity, and when those conditions are fulfilled, and especially some fungal spores, they require some amount of water droplet for the support of absorbing water for starting the digestion of the stored material in order to release energy as well as developing this structure that is named as germ tube. So starting this process, getting ready for this penetration process is the stage of pre-penetration. Now, with the same diagram, we can explain the penetration also, but up to this, now here the drawing is prepared in a way that the consequence of pre-penetration process is illustrated, or the sequence of process is drawn step by step in the same drawing. So the spore, just like a spore like this lying on the plant surface, then developing the germ tube like this. So it is drawn here more clearly. Now, after developing for certain distance, the germ tube is expanded to make a disc-like structure that is adhered, sticked onto the surface of epidermis of the plant organ. Here it is a leaf. So, up to this, the pathogen is getting ready for penetration. That means pre-penetration. Then, now from the lower side of this disc, which is named as appressorium, this disc that is adhered to the plant surface, appressorium, on the lower surface of this disc, a peg-like structure develops, just like the peg in a drawing pen. So that peg, when it is developing, with the firmly attached, sticked, adhered disc, when the disc is adhered to the surface and a structure, a peg is developed towards this direction, it is acting as a drawing pin. With the pressure developed by this appressorium, it is penetrating into this. So that peg-like structure is named as penetration peg. So once it is entering, it starts absorbing the requirements, water, nutrients, and other things required by the fungal pathogen. So once it has entered, it becomes a mycelium already, and that mycelium is now invading into the cells and tissues of the plant. So invading mycelium is now already inside. Now, once the penetration peg has entered, now it has penetrated. Once the penetration peg has penetrated, the pathogen is already inside. It has been penetrated now into the plant tissue. So, the third step has been already completed, penetration. So, what we observed on the screen was the direct penetration using the appressorium. In addition to the penetration with the appressorium, the developing mycelium inside is penetrating, developing into the cells, or in between the cells to the tissue. The organelles of the fungal mycelium that is penetrating into the cells or developing into the tissues of the plant organ, it is named as hostorium. The role of hostorium is absorbing the requirements to the fungus. So this explanation can be illustrated using a diagram like this. Spore, germinated germ tube, developing into a appressorium, lower side of appressorium. So the appressorium is glued, adhered to the surface of the plant organ. Lower side developing penetration peg. Here it is mentioned as infection peg in this book. So then from that infection peg, a mycelium has been developed, and from the mycelium, another type of organ similar to infection peg or penetration peg, a structure called ostrium is developed. So it is playing the role of invader into the cells, absorber of the nutrient and water to the mycelium of the fungus. So this is a diagrammatic view of the germination, and this can be of course observed through electron microscopic pictures like this. So the spore is germinating, developing into the appressorium and making a peg-like structure, penetrate into the cells under this cuticle layer. So these are of course electron microscopic views, and this is of course a drawing. Oidia spores of a fungus develop into a germ tube. The terminal end has been developed as appressorium. Now the story is known to you now. Another drawing, a bit different way of developing the mycelium. Now here the contacted fungus is developing on the surface of the plant organ, and in multiple places the fungal mycelium has produced hostorium, and that hostorium has invaded, or we call invaginate, the membrane of the plant cells. So this may go to the next adjacent plant cell also through the plasma desmata. I hope you can remember crop botany and crop physiology. So the connection between two cells in a plant tissue is facilitated by plasma desmata. So that hole is utilized by some pathogens to move from one plant cell to another plant cell. So these are the methods of penetration. That means entering by force. Whether the plant like it or not, whether there are openings for the pathogen or not, the pathogens are entering like this. We name it as penetration. So the same way of entry is possible by using some openings available on the plants also. Maybe some pathogens they don't like or they can't develop the structures like penetration peg invasion peg, or they are not capable to penetrate by force through the organs or maybe tissues or cells of the plant. But some pathogens, even though they are not capable to produce the penetration structure and use them scientifically, use them with the some biological or physiological methods of interfering with the plant biological process. Some pathogens that are not having any mechanism to by force penetrate, but they are also causing the diseases inside the plant. How do you think? How they enter into the plant if there is no possibility of producing this type of Clostridium, then penetration, pegs, invasive hyphae or invading hyphae. How a pathogen can enter without producing a structure like this? Is there any possibility? Suppose that you want to enter into a plant and you are very small. You don't have any instrument to penetrate the cell wall. How you would like to enter? Will there is a possibility, but you know botany of crops. So how you would enter into a plant to check the plant activities? Or is it impossible for you to enter into a plant? Oh, sorry, I forgot that no listeners, no speakers. So then
Stomata.
little bit louder, please.
So the stomata.
Yes, there are many natural openings, mainly the stomata, lenticels, hydatodes. So if we are microorganisms, we can enter through these stomates, through the lenticels. I hope you can remember these openings according to the studies you had in botany. So these natural openings are helping or facilitating entry of some pathogens who are unable to enter by force, right? So what are the pathogens who are unable to enter? Some fungi, they don't produce penetration pegs, invasive mycelia, Clostridium, but they enter through these natural openings. So guttation water droplets developed near hydatodes, they help spore to absorb water, germinate like seed germination by plant, and they enter into the host tissue like this. So without any trouble, they can enter. Then we mentioned through the stomates, through lenticell hydrathodes, and of course, not only that, even the wounds caused by various factors can be entry points for some pathogens. So, on the plants, there can be some wounds created by wind breakages or rubbing of plant organs parts on each other due to wind or other factors. Then animal feeding. What type of animal? The small animals like insects, when they are feeding, they pierce the cell wall, peel off the fruits, stems, and through those damages caused by insects, the pathogens can enter. Not only that, we do some practices like pruning, grafting. So these cut opening places, surfaces are good entry points for some pathogens. Then during harvesting, we make openings. The surface of this petiole is possible for a possible entry point for some of the pathogens. So how this fruit stalk or petiole become entry point? So when we do harvesting like this, it is same like doing pruning using an instrument like this. Here we cut the branch, make a cut surface. Here, if the fruit is fallen naturally as the senescence process, then there will be a senescence layer developed, detach the stalk from the plant. But that layer is not developed if we harvest. So we do harvesting. Rarely we wait till the fruit is fallen on the ground due to various reasons. Right, okay. Now, from the surface, from the cut surface also, they penetrate by developing those structures like penetration pegs, invasive mycelium, invading pegs like that. So, according to your information you are having about the plant disease management, maybe post-harvest disease management, or management of diseases during the crop cultivation, maybe you can think about the prevention of this penetration also. How to prevent? Suppose that this fruit stalk or the simply we say attachment fruit stalk, if these are harvested by us. Or maybe now, if it is a fruit, naturally pollen or natural senescence is happened. Then on this surface, there will be a senescence layer. But so that layer will be a certain barrier for entering by some other organism. Right? But if this is a fruit harvested by us before the senescence layer is developed, then this rootstock cut surface or surface created by our whatever the detachment is. Now the senescence layer is a layer with some cells with some deposits. So the senescence layer cells are having some thickness that will be difficult for the penetration by a pathogen like fungi. Some are penetrating. But when this is the situation, then the pathogen can land on this surface and start possibilities of I would like to just ask from you. So right. So then what will happen if now I'll draw a relatively larger spore, and if it is germinating, there is no senescence layer, a germ tube start to penetrate like this, and it will go through the fruit stalk to the mesocarp of the fruit, because the mesocarp is having the stored food. Now, I'm not going to talk about all the methods of penetration, how to prevent. So I hope you will read. I know that you will be a bit reluctant to talk about the controlling methods if I am asking. But how can we suggest to a farmer to avoid this penetration of fungal pathogens through this fruit stalk when you do harvesting? What are the possible suggestions? Maybe the students who are working in the Department of Agriculture can suggest more methods than me, because they are having this expertise of instructing farmers how to do harvest. Their recommendations are very important. So those who previously helped me, can you please help me now as well? Number 141. or I can remember Roshan, then I can remember last week there was a number 32. I don't know whether he or she is here today. Number 32. Right. So what to do? Now, if the germ tube is developing like this, going through the stalk, entering to the mesocarp. Now, until it reach a tissue or cell that gives some stored food to the fungus. Fungus is using the stored food in the spore, but it is a very small quantity. That quantity will be finished very quickly. If it is finished before the penetration peg, infectious peg is absorbing nutrients, the germ tube and the spore will be dying. So then we can't reduce the stored food quantity, but we can have a long way for the fungus to go through this stalk. How we can do extension of the penetration passage? If we can harvest the fruit with a long, relatively longer fruit stalk, then the germination, after starting germination, the penetration through the stalk, if it takes long time to reach here, during that period, maybe the stored food in the spore will be depleted, finished, and there will be, difficulties for the fungus to survive until the germ tube is reaching the required tissue. Right. Okay. So, yes, anything you want?
We can wash the latex from the harvested fruits.
So, by removing latex, yes, it is a recommendation given. So, how it prevent the penetration?
Fungal maybe can survive on the latex.
Yes. So, it is recommended to wash this latex on the surface and have a longer stalk. So, with the remaining latex, which is a sugary exudate, which is a rich source of nutrients for the pathogens. So, if the latex is there, the fungus is initially growing on the surface, absorbing latex, the nutrients in the latex, and that means on the surface here with the latex, the fungus is having substrate for survival, and from the substrate they absorb nutrient. The latex is the substrate, and so during a certain time period, fungus will be already developing into a mycelium, and then the mycelium can provide resources for developing germ tubes. Even maybe the fungus will be growing here, and some mycelia are going through the fruit stalk to penetrate through this cut end. So that is possible. And of course, it is a recommendation by the Department of Agriculture, or not only here, even in in other countries also, that recommendation is there. So harvesting with stalk, longer stalk, and if the product harvested part is having latex or any kind of exudate, washing it also strongly recommended. So thank you for that suggestion. When the pathogens are penetrating, there can be two types of
We can use suitable equipment for harvesting fruit and harvest crops.
so instrument that with minimum damage to the tissues. I hope you want to say like that, isn't it?
Yes.
Yeah, and also we can use clean instruments, maybe sterilized instruments, disinfected instruments, because from one place to another place, there can be a dissemination of pathogens with the instruments also. So there are different types of penetration methods with the intercellular or intracellular penetration. Once they enter into the host, they start further development with the process of infection. That means entering into the host and persist in the host, right? So entering, we know that. Persist means remaining for a certain time period. It is not just remaining, active presence, active remaining in the host. So that means the entered pathogen is now developing further, starting development of not only germ tube or hostoриum, other structures required for their reproduction. So for all those activities, they need resources. From where they receive the resources? Resources are received, taken from the host plant. So as a result of that, entering, persisting with the absorption of resources. For the absorption, they release enzymes, toxins to kill cells and tissues of the host plant tissue. So those toxins can damage host plant cells, tissues. It may kill the cells and tissues. And also, now at this time, the plant is also, if it is a plant with some strength, genes to develop some defense mechanisms, the plant and the pathogen both are developing symptoms. So symptoms are now developed, but we know that pathogen has entered some times ago. But the symptoms are developed during this process of their activities targeting the persistence inside the host. So the period between the starting of the infection with the entry of the pathogen until the symptoms are developed, that period we mention as incubation period. So on these potato plants, now only we are observing a symptom of a disease. but probably the pathogen has entered to the host sometimes back. So during this period, until these leaves are getting this symptom, the period is mentioned as incubation period. So the symptoms developed like this. Are developing not instantly. It takes some time. And depending on the places of or tissues of the host through which the pathogen is growing, there are different types of infection we can consider. Some pathogens are developing on the surface, so their development is superficial. So if the superficial colonies are developed on the surface, we name that development as ectoparasite. So the powdery mildew like this, then the oidium leaf disease in rubber, you heard about that. The same pathogen is causing disease development on rambutan like this. So surface ectoparasite. Some are developing under the cuticular layer. If the infection is at epidermal cell layer, we consider it as subcuticular infection. So another example, the black spot of roses. Then some pathogens, they are infecting mesophyll and parenchyma tissues. Then we name them as mesophyll or parenchyma infections. Now, it is essential to understand where the infection is going on, because based on that, you have to select the treatment methods, right? So not only the chemical treatments, even the other methods are also better to be used with the identification of type of infection. Because if it is a superficial or ectoparasitic infection, then some actions that are taken with the active action on the surface will be sufficient enough. But if the developments are inside, like this, mesophyll or parenchyma infections, then our action should be penetrating into these tissues as well. Then some infections are more deeper penetrating. They are developing into the vascular bundles, vascular infections. So wilting will be the symptom. Wilting means droopiness of the leaves like this in the worst condition or later at the end of the infection process. The plants will be totally destroyed like this. So wilting, this can be caused by lack of water also. But when the vascular bundles, especially xylem bundles, are blocked by a pathogen, the water supply, transpiration is blocked. Therefore, the symptoms like this will be possible. So, some pathogens, they develop the symptom throughout the whole plant. That can be possible by pathogens who are capable to go everywhere in the plant. So the viruses are capable to that. Why? They are the smallest group. So a virus can penetrate into any part of the plant if there is an infection. Even now, see, this is the mosaic symptom caused by tobacco mosaic virus on tobacco plant. So here the symptoms are on the leaves. The leaves are infected, and the infected leaves having less photosynthesis. We all know that. And also, if a sucking pest is sucking sap from these leaves and moving into a healthy plant, they will transmit the disease to the healthy plants also, right? So, this type of a pathogen can go. Now, when the viruses are multiplied, their concentration, their number will be higher. So they will be gradually moving through the cell wall connections. I mentioned about them earlier, the plasmadesmata, and also through the vascular tissues. One of the major character of viruses are they are surviving only in the living cells. So only a short time period they can survive on the maybe exudates like latex or cell sap, we call juice of the plant, but not for a longer duration. If the cell sap or the when we prepare we prepare or when the plant is having some exudate with live cells, so inside the live cells, the viruses can survive unless otherwise the pathogenic viruses are unable to survive for a longer duration. So, systemic infections. If this plant is now flowering and producing seeds, the newly developed seeds will be most probably contaminated with the virus inside. So now the infection is going on in the plant at different levels, as we mentioned. After infecting the plant and reaching the maximum possible development inside the host, then the pathogen is starting growth and reproduction and colonization. Colonization means now the pathogen is already dominating on the host. So now the host is already, how can you say, weakened, maybe defeated in the fight. So now the pathogen is producing higher number of reproductive organs, materials, and just like plants producing seeds, fungi, bacteria, they are also multiplying. Only fungi are producing spores. Bacteria are actually they are multiplying. We call binary fission, dividing into two. So that is the way of reproduction by viruses. Then viruses and bacteria. So they are multiplying their particles. So anyway, whatever the method they use, they produce huge numbers. Bacteria and viruses viroids, phytoplasma, they produce around bacterial life cycle is completed maybe around 15 to 20 minutes. So within 20 minutes, they are divided into two. See how faster they produce their progenies. So therefore, huge numbers are produced. So out of these, even if a very low percentage is surviving, there can be a huge risk for the host plants, right? So they produce these propagules and now they are ready for dissemination. So these produced spores, propagules of the pathogen can be disseminated through the wind. So wind will carry the spores as well as the infected plants with their aroma, the plant aroma, or due to the induction of production of some plant exudates by the pathogen, it attracts the sucking insects, the plant consumers. They are coming, sucking the cell sap. During that, if they release the enzymes and hormones to digest, mainly the enzymes, and then absorb the inner material. So with the cell sap, they are absorbing. Virus particles can be also absorbed, and when they move into a new plant, they will first release the saliva. With the saliva, the virus particles can be released to the new place of the same plant or new host plants in somewhere else. So through the wind, there is a possibility of dissemination of the pathogens. And by us, with the instruments we are using, with the clothings we are wearing, there is a possibility of dissemination of the pathogen. So I didn't ask with the last few steps because it was very difficult to get an argument, answer, or support from you. Only a few are actually, I repeating, it is supporting me as well as maybe more than that, supporting yourself also. So if we handle the diseased plants, better to handle with care by cleaning the instrument, if it is possible, from each plant to another plant. Better to clean the instrument so there will be no transmission of viral diseases especially. So once these developed propagules are disseminated in a suitable place, the pathogen will survive. Sometimes the pathogens, once they are disseminated, they start directly the next cycle on another plant, starting from the inoculation and pre-penetration, penetration, so on. On the other hand, the plants that are having a disease like this, downy mildew, this type of Sclerotium disease, some bruises, compressions on apples, a disease caused by a fungus, so infected these leaves. So if these leaves are fallen on the ground, so it is a survival place for pathogens, infected fruits, leaves, virus infected wheat plants or other plants. So all these are survival places of the pathogen. So from the survival places, they can come back to start with the inoculation. So we have gone through the process of disease development, and I mentioned that we are talking about the disease development process with the aim of controlling the plant diseases. So if you are now in touch with the session, then you may now think, at least now, at the end of your so in each and every step, we can interfere with the development of disease. So we were trying to mention how to prevent the inoculation. So now again, I would like to request you to suggest any method that can, any method of plant disease management that can reduce the risk of infection by that pathogen in the maybe in near future or in a new place. We can use that. So while you are thinking about that, I would like to mention now this is a structure produced by a fungus called Sclerotium. So their fruiting body, where the spores are produced, is named as Sclerotium. That is why their genus name is also given like that. So I would like to mention another term used in plant pathology. Now we can see that some fungal mycelia are grown here. So this is a crop residue or decaying plant part. These types of residues are the debris with a higher risk of reinfection. So then they are thinking about managing methods of this pathogen more efficiently, because when this type of sclerotia is produced by a fungus, if it is fallen on the ground, it can live a longer duration. So it is very important to understand what type of propagation methods are used by each and every plant pathogen. Right, okay. Any method of disease management, how to interfere the disease development process of different pathogen groups, especially the disease management during the disease development process steps. How to control diseases at the step of infection, inoculation first, then penetration, pre-penetration, no penetration. If you manage the diseases, yes, you are achieving a healthy crop, healthy fruits, healthy plants. If not, there can be the plants with disease symptoms. So maybe for the fifth time, I am telling you what are the possible disease management preventive methods that can be used to interfere, control each and every step of disease development process. So if we know the disease development by fungi, it will be easy for understand other pathogen groups. So other pathogen groups infections are also explained in the module as well. These steps also mentioned there. Right. Any questions? We will continue with the disease management maybe. So without maybe consideration of this disease development process, I mean, without repeating some principles of disease management. Okay. Anyway, that is the next topic. But about this, any questions you have regarding the disease development process? Anything you want to clarify about what we have discussed up to now? Right. Okay. Then if you don't have any questions, we will have the second break, another 15 minutes. So maybe some of you would like to have something as a meal or lunch anyway, but I am going to have the similar pattern of the sessions like the last Saturday. So take a break and continue interactions, listening, speaking, and yes, I saw some of your messages, but unfortunately, it was too late when I was having the attention given to the chat box. So I always prefer to hear your voice, then you are more active, and the responses will be more active, so interactions are more clear, prominent. Right, anyway, you are now having a break. I'll also take a break. Try to be connected because reconnection may be difficult due to this poor connectivity time. all these are topics in the module as well. So I'm repeating an answer I gave to one of your questions raised last day regarding the content. So I'm trying to give a clarification on the core material, the most, how can you say, the fundamental things in the module book. So once we complete today's as well, maybe as you were interested, some of you, regarding the questions for reference and for revision, I'll put a special, separate section on LMS to give you some questions to think. Even in the discussion, I am raising many questions related with reference, revisions, to make you think more about the important aspects. So try to use those the thoughts we are having in our contact session topics in your mind. So then at least now the best thing to get the maximum benefit out of those questions, the best thing is to actively comment on those things while in the session. Then I can understand also how deep you are thinking, what are the problems you are having when you are thinking about those questions. But if you are not responding, I am not in a position to guide you or make you aware about the difficult things for you. So then another thing is I can feel that some of you who are here are a bit more interested on the discussions, responding to me. So I invite you, if you are free, you can join tomorrow as well. I saw some of you were joining the Sunday group even last weekend. So I felt last weekend that in Sunday group, more members, not all of them, but a higher number of students were active in the discussions. So the active students here, they are, I mean, cordially invited to the tomorrow session as well. Of course, the same topics, but with the discussions they initiate, maybe we can think about some additional aspects of the pest management protection as well, right? So, okay, we will move into the disease management. Another important thing is, now when we were talking about the pest management methods last day, I mentioned that those methods are equally valid for management of diseases, weeds and other organisms as well. You can see that if you are reading the module also. Then today's these topics, of course, the earlier one about the disease development process, it is related with the diseases, that is true. But this one when we are talking about plant disease management, and we are going to talk about the principles behind the management. So those principles are valid for the other groups of pests as well. So the integrated management, the methods, principles, all are equally valid for the other groups. So you can see in the module it is written as common for all groups of pest. Right. Okay, then the plant diseases. We studied about the development process of these types of diseases. So all these, these spots on the leaves, fruits, and the yellowing, if it is caused by a virus. So regarding the virus disease development process, you can read a lot in the module as well. So now these diseases developed according to the same steps, starting from inoculation until survival. So these leaf diseases in banana or any disease. So what we want to do is management of those diseases.
Sir, we can't hear.
Sir,
Yes, I understood. I understood. So major principles of disease management, they are based on the disease triangle, the classical explanation of the disease development, given in all kinds of literature sources on plant pathology, disease management, because if we want to manage a disease successfully, we need to modify the environment, we have to take actions on the pathogen, we have to take actions on the host. That means all three aspects are important. So dealing with only one component may not be successfully controlling our diseases, plant diseases. So because of that, all plant disease management principles are based on this diagram. I am not just mentioning about it because I am having that diagram. You can see by your own if you are reading the classic examples of plant pathology books, or maybe in an online literature source. Also, you can find this disease triangle explaining about the plant disease management. So, when we think about the disease management, we need to start with the correct identification. So, as I mentioned, the disease triangle is also a principal aspect of plant disease management. So, when you say diagnosis, usually we think that we are diagnosing what is this, the name of this disease. Of course, that is important. And knowing the name or knowing the disease by the name, it gives a lot of information. That is true. But not only that, when you say you have diagnosed, identified a disease, it is identifying the causal organism. What is the organism causing this disease? And then what are the symptoms developed by that causal organism? Or maybe we are identifying the disease by the symptoms developed, isn't it? The first ever identification, first ever diagnosis will be given by the identification of the symptoms. Not only the identification or knowing the symptoms and the name of the pathogen causal organism, there are many things needed behind this diagnosis or identification. When you say identification, with the identification of the organism, we need to identify what are the major biological aspects of that causal organism. What type of organism is that? Where it starts its life cycle. So just like identifying the life cycle of an insect, life cycle of a fungus, life cycle of a bacteria, virus, you may think what type of a life cycle can be with the virus, which is only a protein capsule and a nucleic acid molecule, RNA. Yes, that is true, they are very simple, but even a virus is having a cycle of its life, and it is creating a disease cycle. So the life cycle of the pathogen should be studied, identified, in order to identify the disease cycle also. So we were talking about the disease cycle. So we need to know the information of the disease cycle. Disease cycle cannot be studied or understood if you don't know the pathogen biology, pathogen life cycle. So because of that, when you say diagnosis, you mean a lot, right? So it gives us where the pathogen survive, how it can come into the contact with the host or inoculate, how it can enter or penetrate into the host cells, tissues, how it can start infection, growth and development like that. So those things are necessary to be known before we start thinking about the management plan of the disease. Right. So we identified all those things. Then the next thing is prognosis, forecast. Thinking what is going to happen on our field with this pathogen. Pathogen we identified. So we saw this symptom, identified. So now we have to think what will happen in near future. A forecast. So we have to anticipate correctly, not just guessing something, anticipating what would happen if there is a disease development process going on in near future. So now with the basic information we are having about the disease causal organism, its biology, then we can think about the disease development cycle. So maybe we have a healthy crop at the first glance, but maybe we have identified a pathogen on one or a few plants in my field. So I have to have a forecast, a prognosis about what are the things going to happen on my field. So disease cycle, the possible cycle. Then if that cycle is going on, what are the possible damages to my crop? Because based on the damages, I am taking the decision on controlling whether the control is necessary. If it is necessary, what to do. Then we need to know the possibilities of epidemics of the identified disease. So don't think that finding possibilities or thinking about epidemics is something too early now. We don't have a disease even, and we are thinking about epidemics. No. An epidemic can be occurred at any time. It may happen even starting from a single plant that is infected. How an infection on a single plant may become an epidemic, because when you have an epidemic, maybe you are going to have a disaster based on plant disease. Then when you say epidemics, prognosis on epidemics means we need to have a forecast whether there is a possibility of having an epidemic. Preparing for an epidemic means a different thing compared to the managing a disease at normal level. Then we need to know where the disease causing organism is surviving in our ecosystem. So, knowing these things will be making us very efficient, comfortable with the management of these plant diseases. So now we have two major important requirements fulfilled: diagnosis and prognosis. Then going for the management. For the management, we need to consider what are the best control strategies and tactics that should be applied in this disease management. So we can go through the possible strategies and techniques and tactics, strategies. So out of them, the best selection should be done. Because for each and every situation, according to our diagnosis and prognosis, we have to select the most suitable ones. Once we select the control strategies and tactics, then of course the next thing is applying those strategies rationally. Rationally means it is not just applying everything coming into our mind as suitable strategies. No, the tactic should be applied with a justification, right? So for an example, maybe according to the information we are having, it is necessary to, it is possible, sorry, not the necessary, it is possible to apply seed treatments before establishing the crop. Then in recommendations, it is written that after emergence of the seedlings, once a week, there is a fungicide or other chemical recommended for application. Then it is recommended to have once in a while application of maybe some nutrient boosters. So all those are good things, maybe, but whether it is necessary for applying all the possible things. So in many crop recommendations, especially given to the outgrowers sometimes, if you go through them, they are of course all possible methods of controlling the diseases. And whether it is necessary or not, we have to decide. Some of the management aspects of the diseases are included in cropping system management also. So now when we select the control strategies and tactics, the first thing we need to do is whether there is a rationale for application, and then whether these strategies can be incorporated into the cropping system management. For an example, you have suggested in the morning about use of clean planting material. It can be incorporated in our cropping system management as well. Then a similar action, maintenance of the phytosanitation as a control strategy of some fungal diseases. Is it possible to include that in normal routine cropping system management? Because if we include them in the cropping system management, then we know that the normal management activities will be having some effect on our disease management also, right? Now here I can refer to the thing I have mentioned at the beginning. Now these are equally valid with all groups of pests, insects, pathogens, and the weeds also. When we incorporate the strategies into the cropping system management, it facilitates managing all groups of pests in a combined effect. So that is very good for us, considering the cost of operations and outcome of the management as well. Then whatever the strategies, tactics we apply into the cropping system management or in disease management or pest management, like any other management system, we have to have a very good monitoring program, an efficient, regular monitoring program on the pest and disease management, pest, disease and weed management. So, what are the aspects of management we are having? What kind of strategies that are possible to be applied? What are the major principal strategies to be applied in disease management and other groups of pest management? So, the strategies can be categorized into major three categories. One set of strategies are suitable for application before the attack, before the infection by the pathogen, or before the attack by the pest, or before the invasion by the weeds. So, the strategies that are recommended or considered as strategies suitable for application before infection or attack, they are exclusion, avoidance, eradication, and protection, right? So we are going to have details in a few minutes, but just one thing regarding this strategy. Now, you all know the meaning of the word eradication. Eradication means removing the pest or the pathogen completely. And usually when you say eradication, the idea coming into our mind is you are doing something to remove, destroy, when it is already available, isn't it? Now, for an example, these days, we are thinking about the eradication of the dengue fever, mosquito problem, and we have the problem. And we had a program on eradication of filaria from Sri Lanka. And according to the health authorities, it seems that we are successful in the eradication of filaria. But with the eradication of dengue fever, we are not successful. Once in a while, once in maybe when there is a rainy season starts, always in Sri Lanka, we are having the talks about dengue fever, how to control mosquitoes, and we have mosquito control programs. And of course, a lot of things are done, lot of things are discussed, but not successful, right? So now the question is, if there is, now here we are considering strategies before infection. If we are considering the actions taken before infection, that means there is no disease, no attack by an insect. So how can we eradicate something that is not available, isn't it? Is it true? Now, in plant disease management or plant pest management, we are thinking about taking some actions so that there will be no pest attack. By that way, we are eradicating before it comes. That is why it is given here as strategy before infection. Right, okay. So if we could not do something before the infection, then we can think about some kind of a therapeutic action when there is an attack already occurred. So by applying some chemicals or physical action, we can try to have some therapy, curing of a plant disease. So once there is an infection started, then we can't say that we are going to have strategies before infection, because infection is already there. Now what we can do is having some strategy of the curing therapy, right? So if we take those principal strategies one by one, little bit of details, maybe that will be a good thing to understand the major principles of plant disease management. So the first strategy we have mentioned, exclusion. What are the methods of exclusion? One of the most efficient and important methods of pest or disease or weed exclusion is quarantine, right? I think you all know about the meaning of exclusion. Exclusion means keeping something away from, maybe from a place, from a object, from a territory, like that. So here we are thinking about keeping the pest or diseases away from our cropping system. So quarantine. See the idea given, the policies and programs to prevent introduction of pathogens. Not only the pathogens, this may be policies and programs to prevent introduction of insects, introduction of weeds or pathogens, nematodes, like that. So plant quarantine. what are the ideas coming into your mind? If you have read the information in the module chapter where the quarantine is a topic, and I mentioned last day that about quarantine you can read a lot. The practical things can be read in the agriculture department website. So they are discussing all the details about the plant quarantine in Sri Lanka. So in our country, the authority on plant quarantine is vested on the National Plant Quarantine Service under the Department of Agriculture, and they are having a collaborative program actions taken with the Sri Lanka Customs, Department of Immigration and Immigration also sometimes. Right. Anyway, I think there are few students who are from the Department of Agriculture, and maybe the students who are working in private sector but having exposure to the quarantine procedures according to their workplace, or maybe there are students who are having the experience of having done quarantine procedures in maybe at least in one occasion in our life. So here there is a definition given, but what are the things coming into your mind when you are reading something about quarantine? What do you mean by plant quarantine? Generally, what is quarantine? Can somebody explain little bit about the quarantine procedures known to you? Have you applied quarantine actions? Have you ever experienced plant quarantine in your workplace or anywhere? Maybe you have the experience of being quarantined during the 2020, the COVID-19 pandemic period. So it is a major method of exclusion of pests and diseases or weeds. Roshan, any idea? Right, okay, then of course you are going to read the chapter on plant quarantine in the module. I'll put the request or question in a different way. Now, when you say quarantine, it is something we carry out during the trade or exchange of plant material in between two countries or two people residing in different countries. Can it be a quarantine procedures carried out inside the country, inside the province, or inside the farm itself? Is there any possibility?
For Valigama wilt,
For Valigama wilt, we apply quarantine. So that means quarantine to prevent entry from other countries into Sri Lanka. With the Valigama wilt, we do some quarantine actions. So are they targeting the introduction of Valigama wilt from some other countries into Sri Lanka? Am I correct?
no sir, province to province.
Yes. So that means I maybe province to province or district to district. Now the name itself gives the idea that it is occurring somewhere at Veligama or in southern province. Actually, it was reported initially from Veligama area. That is why the name is given. So probably you may have some information about the pathogen. What is the maybe the group of the pathogen? Maybe not exactly the name. What type of pathogen is causing the Valigama wilt? A fungus, bacteria, a virus? It is a phytoplasma. And it is somewhere in the taxonomy, it is somewhere between bacteria and virus. So phytoplasmas are causing many plant diseases. So when an infection occurred by a phytoplasma in a plant or any other organism, it is not possible to treat or cure once the infection started. If the disease started in a coconut plant, very good example you gave. That is why I am taking a time to explain the plant quarantine with that example. So now the first thing we would like to keep in our mind is Valigama wilt is a disease we found or we diagnosed in our coconut plantations relatively recently. So we had no observations recorded, information records about the Valigama wilt. Maybe it was first reported, I can't remember exactly. If you know you can correct me. I think it was reported somewhere around, not a very recent, around 15 years ago, maybe, right? So I can remember I had two external degree, I mean, like you, I have two, my research students, both of them were from working, one was from CRI, Coconut Research Institute. He was actually a person attached at that time to the Veligama wilt control program. And there was another one from CCB, a student working in a project of CCB or CRI in a control program of Veligama wilt in Gold district, right? So a very good example means quarantine. Usually we think that we are having quarantine actions against the diseases or pest or whatever the pest problem occurring for the first time in the ecosystem. So we wanted to prevent our country from COVID-19 by having quarantine. So similarly, as we had no situation of having Veligama wilt or wilt of coconut caused by phytoplasma in our country, we have common pest in the coconut. There are many diseases, and coconut black beetle is a common pest. It is everywhere. Coconut red weevil is there. And of course, coconut red mite, it was not in Sri Lanka, but now it is a common one. When the wilt caused by phytoplasma first reported from Veligama, it was named as Veligama wilt. And as it is not available in other areas, we thought, we means actually the responsible authorities from the relevant ministry, Coconut Research Institute, CCB, and anyway, all the related people thought better to exclude the wilt, phytoplasma wilt, recorded in Veligama area, keep it excluded from other areas. So we would like to have maybe Kalutara excluded with Veligama wilt. We would like to have coconut triangle excluded with Veligama wilt. So we know that if it is disseminated by the factors, now we know that there are many methods of dissemination of the plant diseases. Maybe some vectors are transmitting the disease, maybe through the wind or other environmental or natural factors for which we are unable to take any actions to prevent wind blowing or flying insects, right? But we can try to prevent the spread by having or taking some legal actions. So, for an example, it is restricted to transport the coconuts harvested from the infected areas. Then, as a plant infected with the coconut wilt, Veligama wilt, cannot be treated and cured, it is strongly recommended to uproot the plant burn and bury in the deep pits. So there will be no spread. Then as the coconut plant is highly valuable these days, at that time, it was decided to compensate the cost of a coconut plant by paying rupees 10,000. But now people who are having coconut wilt disease affected plants, they don't like to remove the plant because they think that somehow they can cure it. Keeping the plant will give them more income. So they are secretly keeping those plants. But from that plant, the sucking insects or other methods transmitting the disease. So legal actions are taken based on the Plant Quarantine Act and Act of Plant Protection and the acts of establishment of CCB. They have the power to forcefully destroy the infected plant, the plants with quarantine pest. Quarantine pest means pest problems that are not available commonly in our country. Then if you read the information on plant quarantine in our module or in the website of the Department of Agriculture, there are categories of organisms quarantine. Quarantine pest, restricted pest, and even a serious pathogen that is available in our country. But if it is coming with some other plant material or inputs to agriculture sector, that material can be restricted or prohibited to entry into Sri Lanka because some new species, new types of the pathogen can be entering into Sri Lanka, right? Okay, coming back to quarantine of valley gummer wilt disease, right? Please don't think that I am explaining or having a session on valley gummer wilt. No, I am explaining plant quarantine based on valley gummer wilt. So, according to the quarantine concept, it is a group of methods based on legal methods mainly, based on rules and regulations. So, according to the quarantine law, the planting material, plant product, or inputs of agriculture should not be allowed to be transported from a quarantined area. On the other hand, if you have a risk of introduction of any serious pest into your crop, you can have rules and regulations to prevent the entry of that pest into your system, right? That means plant quarantine is not only a system of exclusion of the diseases from a country or from a province, as you mentioned. You can have quarantine rules and regulations applied in your own cropping system. For an example, same wilt disease in coconut. Maybe in your plantation, there are few coconut plants infected with valley gummer wilt disease. But you are in your own plantation, right? And you think, my plantation, my coconut plant, why should I bother about having quarantine regulations, uprooting my coconut plant in a good stage of bearing? So I'll keep them secretly and avoid removal and destroyal. By doing that, you are having the serious risk of infecting, having the secondary infection, now you know better than me. So secondary infection of your other healthy plants within your own plantation. So quarantine actions should be taken to prevent the spread of the coconut wilt disease from your plants into other plants of your own plantation, right? So I wanted to highlight that as well. What is that? Quarantine procedures are important not only as procedures to prevent, exclude diseases coming from another country, another province, another district, or another plantation. It is a system of preventing spreading of the diseases or excluding diseases from one place to another place of your own cropping system. Even in a home garden, you can have quarantine procedures applied, right? So keep that in your mind, and with that concept in your mind, study about quarantine procedures. Then when you are exporting plant products or importing plant products, especially for use as inputs into our cropping systems, and when you are exporting the live plant material, not processed plant material, to other countries are very strict on quarantine certificates issued by National Plant Quarantine System or National Plant Quarantine Service. So by having a quarantine certificate only, you can export plant products legally. If you do it illegally, then it is up to you. There is a risk of rejection, returning, and many other problems created by illegal exports. But phytosanitation is having a very close connection with the plant quarantine as well. One connection is that when you are exporting or importing, you need to have phytosanitary certificate for some products, plant material as well. So quarantine certification and phytosanitary certifications both are issued by the same institution in Sri Lanka, National Plant Quarantine Service. So why is that? Because phytosanitation is important method of excluding a pest problem. Now here it is given as, when you say phytosanitation, using pathogen or disease or pest-free planting material. Why? when you bring a planting material with pest or diseases, that means you are bringing the pest problem. But if you use disease pest-free planting material, that means you are keeping the pest away from your cropping system. You are excluding the pest away from your cropping system. But when you say phytosanitation, it is not only use of pest-free planting material. Phytosanitation means, what is the meaning of sanitation? Keeping hygienic condition, clean condition. What do you mean by clean condition in a cropping system? Clean condition in a cropping system or phytosanitary condition in a cropping system means having a cropping system without sources of infection, sources of inoculum, or sources of infestation. So, if your cropping system is free of plant debris with pathogens or pests, you have a phytosanitary condition in your field. Then it means not only plant debris, it may be the plant weeds, crop weeds, because weeds are alternate host of other pests, right? So, to keep the pest away from your cropping system, you need to have good phytosanitary conditions, right? So, if somebody is asking you, what do you mean by phytosanitation? Then you need to explain him not only about pest-free planting material, maybe before that you need to have inoculum-free, source of inoculum-free, sources of infestation-free. What do you mean by sources of infestation? When you say sources of infection, it means the sources of inoculum that can cause infection. But there are some sources where the other pests, not only pathogens, other pests that can come into the crop. For an example, same crop, coconut plantations. In coconut plantations, the coconut plant debris are places of egg laying by coconut black beetle female. So, the larvae is feeding on coconut plant debris and they are the sources of infestation. Infestation means attacked by an animal pest, not a pathogen. Then weed plants, if they are keeping some insect pest, weed plants become sources of infestation. So then having phytosanitation and plant quarantine, you keep some pest away from your cropping system. Right. Then regarding the cultural practices, actually about the cultural techniques of pest management, we had a lengthy discussion last week. So what I have to add is that some cultural practices are keeping pests away from our cropping system. What are the cultural practices? If I repeat the same, it may be monotonous. What do you mean by cultural techniques, cultural practices of crop protection? According to your idea, how can you define or explain cultural techniques of pest management, which we have discussed last week during the same time, in between, yes, 2 o'clock 2:30, we were talking about cultural practices, cultural methods of pest management. The information we have discussed last week. Do we have anybody who attended last week contact sessions? No. Maybe at least one.
Crop rotation, field sanitation,
Okay.
resistant varieties grown,
Yes, thank you.
intercropping.
So that means what you are mentioning is they are normal cultural practices, cultural techniques applied for crop establishment and maintenance. So the same practices are useful for management of pest. So by doing some of those actions, we are keeping some pest away from our cropping system. So perfectly correct. You mentioned that use of crop rotation. By rotating the crop, we are changing the place of cultivation, the land plot, and then when we change the crop, the residues of that crop, for an example, my crop of Solanaceous family, if it was infected by the bacterial wilt disease, so the bacterial pathogen infected my crop root system is available in the soil. Can I remove all infected roots from the soil? No, I can't. So if I am growing the same family crop, not only the same crop, even if I had tomato bacterial wilt this season, if I grow brinjal, maybe capsicum on the same field, there is a possibility of having the same pathogen attacking the next crop. So better to change the crop in a way that the next crop is having non-host condition for the bacterial wilt pathogen. So we were talking about the resistance last week. So if I'm growing cowpea on that field, so the remaining bacteria in the soil, they are unable to attack my cowpea plant. And if I have rotating fields, in the field where I grow cowpea earlier, now I can grow solanaceous crop. So if I return solanaceous crops into the same field, maybe after 5 years, that is the best time. But at least for more than 2 years, if I can keep the crop away, then it is an indirect way of excluding bacterial wilt from my crop, right? Okay, I'm not going to repeat the information we discussed about the cultural methods of pest management last week. I'll just request you to think about the revision of that material by your own. But if you want to ask any question, in relation to the cultural practices as methods of exclusion, you are welcome. Then we'll move into, after having a long discussion on exclusion methods that are very important, right? Why I took some more time. Now, excluding means keep the pest away from the cropping system. That is the best thing. If you have kept the pest away from your cropping system, it is better than treating the pest when it is available in the cropping system, right? Then, having more time to discuss the best thing, we will move into the other one, avoiding the pest. One is that we can establish our crop in a place when the inoculum is inactive or when the inoculum levels are low, maybe due to the environmental conditions or some ecosystem conditions we are having. The inoculum of a particular disease will be low, maybe due to the temperature changes or humidity change. Then if we can identify such a time, so it is better to establish our crop in such time period. Then some diseases are available in certain areas only. So for an example, the example given by you, nowadays, at present, our experts or responsible people, responsible scientists and other officers in coconut sector, they were successful in keeping the Valley-Gumma wilt disease confined to southern province only up to now. And even in southern province, in certain areas, they are conducting the varietal trials to test resistant varieties against the coconut wilt. If you are from the coconut related institute, you will know that better than me. So somehow, with their effort of keeping coconut diseases, coconut wilt disease caused by phytoplasma in the southern province. So in other areas, there is no threat of growing coconut at present, right? Up to now. So we can avoid coconut wilt disease in other places. So it is a good example of avoiding. Then another method of avoiding the diseases is production of planting material in areas away from commercial production sites to avoid the pathogens. So again, the same crop can be taken as an example. So the breeding experiments and seed planting, the seed production of coconut varieties are carried out in an area confined for seed production. Seed means coconut seed production away from commercial coconut plantations, or there is a designated area called, if I am correctly naming that, Ratmalagara area, to produce coconut planting material. But you can do it in your establishment to avoid the infection from commercial production areas, because In commercial production sites, we are growing high yielding varieties, giving higher inputs for increase of production that will attract many other pests. So because of that, better to keep the planting material production away from commercial production sites. Then, if we could not prevent the infection, prevent the attack, then we can try to eradicate as well. But eradication can be planned well ahead before establishment of the crop also. So, to reduce, to remove or eliminate the inoculum at the sources, of infection available in a particular area, particular plant that is already established. For an example, if you can remember in the morning, we saw a banana leaf disease. So that disease is available on the leaves. So when we see such leaves, especially the older leaves, when they become older and infected, quickly we have to remove and either bury or burn in a distant place. So other than that, other than having phytosanitation as a eradication method, we can do a lot of other methods like crop rotation, then treatment of the inoculum sources, because we do crop rotation and the pathogen or the pest will be losing the host. I think you can understand that easily. So use of certain treatments for eradication of the pest and pathogens, we can do eradication. Then this is an illustration, use of biological control agents. So you have many information about biological control agents in the module. So one is about fungus called trichoderma. So actually this is a diagram only. So we can introduce trichoderma fungus into our cropping system. So that means it is growing on organic matter, and some organic fertilizer producers are incorporating trichoderma cultures into organic fertilizer. So trichoderma, when it is there, if a plant pathogenic fungus is growing, trichoderma is parasitizing on that plant pathogen and it suppresses the plant pathogen. So trichoderma is a biological agent used to control pathogenic fungi. Right. Then the other strategy before attack is protection. So we can think about reducing or eliminating the effectiveness of inoculum. Now you know what is an inoculum. So at the site of infection, we can have a barrier between the inoculum and the pathogen by applying some chemicals. So here we can consider even fungicides or other chemicals that destroy insects also. So we will try to make a chemical barrier between the pathogen and the host. And also non-chemical barriers like having a barrier for the vectors, netting material is possible for protection of the plants from the pest. So I just mentioned about the application of chemicals, treatment of seeds, use of protectant fungicides, developing the resistance by reducing the effectiveness of inoculum. How to reduce the effectiveness of inoculum if we know that inoculum is there? In addition to the applying chemicals, we can expose the inoculum source to sunlight or unfavorable condition for the inoculum. So I just found a photograph of now seed treatment test. When the treatments are carried out, the germination is enhanced. Treated seeds are giving better seedlings, higher germination percentage. So it is a way of protection from the seed-borne pathogens. Then, of course, if we could not take any action before the attack, then there can be some attack by the pathogen. So then we have to take therapeutic action, curing actions. So what to do? Now we have to apply the therapeutic actions by using therapeutic means it can be a chemical treatment, it can be some physical method of treating, like now here you can see an action taken to apply a chemical on the leaves. Then not only that, sometimes small scale propagator type structures can be used to create little bit higher temperature condition to the plants, planting material, so that the pathogen can be destroyed or suppressed, so that the disease will be at lower level. So then maybe as a not for a break, but as a change, as a relaxation, I have a different type of a slide. Maybe you will think, what is the need of this type of a slide on the screen? Think little bit and maybe you can explain the thing I want to discuss with you. So these are photographs of a novel, නවකතා පොතක පිටකවරයක් මේ තියෙන්නේ. Earlier published, so I can't remember exactly the first edition publication date, but this book has been published again and again by different publishers, and it is originally written in English and translated by different translators. I couldn't find the cover page of the translation which I read in my school time. The translator was Mr. A.P. Gunaratna. But here another famous translator, Miss Anula de Silva, or Mrs. Anula de Silva rather. So what is the relationship with this book? Is it just a distraction from the topic plant pathology or plant disease management? Have you heard about this author or the book? course, last week, without a lunch break, I could manage to stop at about around 2:30, but today, due to that technical fault at the Zoom server at our faculty, I couldn't start on time today. You can remember that. So that is why it is a bit late today, but I hope you can tolerate. Now you know what is tolerance and what is resistance. So anyway, expecting the response from you. Number 57, can you, I don't know your name. That is why I'm asking from the number. Maybe Roshan or number 141, who had some frequent responses to me in this Saturday group. Right, okay. If you, okay, then I'll help you in this regard. Now, this story is written based on the one of the commonly referred plant disease epidemic in almost all plant pathology books. When they are talking about the epidemiology of plant diseases, they are mentioning about this story. What is the story? This is actually, this is not a plant pathology book. It is a novel written by this lady, Christine Wilson. She was a resident of Sri Lanka. She is the daughter of Dr. R.L. Spittle. Have you heard about that gentleman? He was a British officer residing in Sri Lanka. He came to Sri Lanka, and because of his attractness to Sri Lanka, he was not a planter, medical doctor. He was having a love to our people. He wrote many novels, so I'm not going to talk about the novels written by Dr. R.L. Spittle. So you please, I think I'm naming it the name of the father correctly. So Christine Wilson, she died relatively late in recent past. I can't remember, in 1980s or 90s. So now in anyway, with the support from the father and with the information collected from the historical reports, this lady has written this novel about the coffee rust disease epidemic was going in the background, right? So she was writing about the change of some sociological behavior, sociological background of some planters family. You all know that British people started coffee plantations first in Sri Lanka, right? But due to the negligence in the maintenance of quarantine regulations, quarantine rules at that time, they have not thought about the quarantine procedures. They have not thought about phytosanitation, and one of the method you have suggested today, they have not considered the importance of the use of pest and disease free planting materials. Some of the planters they have imported, they have maybe brought or bought from some other countries infected planting material, contaminated planting material, or planting material with the inoculum of a rust disease. So because of that, the large number of plantations were destroyed by this disease. So what they had to do at the end, they couldn't prevent the attack. They couldn't do exclusion. They couldn't do protection. They couldn't attend to the eradication. So they had to avoid cultivation of coffee. So because of that, they had to change the crop into tea. So because of this epidemic we had in 1860s in our country, in the pathology books they are writing about this as a disease epidemic that changed economy of one country and drinking habit of another country. What is that? Due to the epidemic of coffee rust, Ceylon planters, at that time we were in Ceylon, our country name was Ceylon. So Ceylon coffee would not be exported. We had to change the plantations into tea plantations. So our major income source, foreign income, was based on coffee. But instead of coffee export, we had to export tea. On the other hand, the British people were drinking Ceylon coffee because it was cheap, because our country was their colony at that time and they were having low cost for coffee because their own planters were here. So British people had to change their drinking habit from coffee to tea, right? So we have changed our economical concepts and they have changed their drinking habit. So what is the importance of that for our today's topic? If we are not keen on the conditions that are developing disease epidemics, then there can be an outbreak and spread of the diseases very quickly. So because of that, it is essential to consider how diseases are increasing over the space in severity or over time. So our scientists are now trying to prevent the spread of coconut wilt into other areas of Sri Lanka from the southern province. Then time to time there are risk of epidemic of diseases, pest. Now we could not prevent the epidemic of coconut red mite. We could not prevent the epidemic of coconut whitefly. We could not prevent the epidemic of Sena caterpillar or fall armyworm epidemic. So you know what were the results. So how to prevent the epidemic of plant diseases? It is based on the disease triangle again. So diseases are created by three components: host, pathogen, and environment. So if we want to prevent the disease epidemic, that means we need to think about the host, the pathogen, and environment. What are the things related with these three components in preventing the diseases? If we know the characters and properties that affect the susceptibility or resistance of the host. Of course, the genetics are important here. Not only that, the environmental conditions are keeping the host resistant or susceptible. So, if the environment is keeping our host resistant, that would be ideal for us. If we know the conditions that are keeping the host resistant, why don't we keep like that? Some conditions may be higher humidity conditions, increased nitrogen doses, or other stresses to the plant may keep them susceptible. So then the disease or pest can be at epidemic level. Similarly, some environmental conditions are keeping the plant pathogens virulent. Virulent means very powerful in attacking the host. If the conditions are making them more abundance means available in higher number in many places. So those conditions should be avoided to avoid the disease epidemic. Then the environmental conditions that enhance the diseases should be avoided. Major concepts in epidemiology is that the different pathogen populations are having different disease cycles. So their requirements may vary. Now we are studying about the requirements of phytoplasmic wilt of coconut. So by studying that, we can try to prevent the epidemic of the coconut wilt disease. If you can remember, in year 2018-19, we were trying to study the fall armyworm requirements. Until we were studying about their requirements, there was an epidemic condition. We requested farmers to avoid cultivation of their preference crops. What were their preference crops? Maize and some other vegetables. So we requested farmers to avoid cultivation of maize in the Yala season, especially Maha and Yala of year 2019. So at the end of 2019 Yala, we could manage the epidemic and inform farmers to start cultivations normally. So studying the requirements of pathogens, behavior of the pest or insects or the weeds, then we can prevent the epidemic conditions because epidemics are very serious. Now take that example from Sri Lanka, Ceylon at that time. Because of the coffee rust epidemic, the planters, they had to abandon coffee. If we had coffee plantations, actually, maybe we could have better coffee plantations than the tea plantations, because the coffee plantations are having more suitable, they are more suitable for the cultivation in upcountry of Sri Lanka. But what to do? They had destroyed their own plantations by negligence of quarantine regulations, right? So not only in Sri Lanka, in other countries, there were disasters related with the disease epidemics. So, one of the leading other example was in Ireland. They had potato disease epidemic, and they have lost millions of people. So you read about it. I am not going to tell the story. So with the consideration of all these disease management strategies and principles, the techniques of pest management, disease management we discussed last Saturday, they are equally valid for today also. So then we can have a healthy crop, healthy plants, and avoiding the plant diseases or plant pest, right? So then I know that you are interested in the disease or pest management in order to face your examination also. So the questions for the preparation for examination will be available on LMS on Monday, or maybe tomorrow. I'll try to put it. So with the consideration of our interactions, our sessions today and tomorrow, I'll decide on the home assignment. And I saw that there was a question regarding the examination. So what type of practical examination will it be? It is not that I am keeping a secret. Honestly, I have not yet decided whether it is a written paper. I have discussed with my colleagues from our department. So it may be still it is open decision. So I'll inform you once I have decided. So you just adhere, or you just adhere to the instructions given in preparation of home assignments and information on LMS regarding the questions. Then definitely you will be having good preparation for the examination, not only practical, but also for the theory part. Then regarding the date of examination, I know that you are aware about the date. You asked me about that last week, but we had the discussion on the examination date last Tuesday, I think. Tuesday or Wednesday. Anyway, now you are aware that it will be on 6th June, Saturday, and we have another additional week. So you will be having more time to prepare for the examination and prepare the home assignment also. But if you have any other question to raise, to explain anything from the contact sessions, you please ask. Any question? And my invitation for those people who were active today to, if possible, it is not compulsory, it is just an invitation, spending time to listen to the same topics again and again. It may not be interesting and monotonous, it may be, so then it is not compulsory. Maybe occasionally, once in a while, you can join and have your inputs, just like today. You can help me to make live discussion tomorrow as well. Otherwise, you can have some other work carried out by you Right, so if you don't have any questions further, I'll wind up for today. Keep in touch with the LMS, and then you will be having the information on the home assignment and the revision questions as well. So why they are not available today or up to now? Because they will be prepared based on our exposure in the contact sessions. Then it will be more fruitful considering the exposure during the contact sessions as well. So it is not just routing questions, routing home assignments. I am planning to have the assignments and the questions to expose you to the materials that were unable to be discussed during the contact sessions. Right. Then if you don't have any other question, yes, there is a question. I don't know why you are not asking verbally. Cultural practices are, yes, non-chemical methods. If you consider chemical application as a cultural practice, then of course, my answer is not valid. But we are considering crop establishment and maintenance practices as cultural practices. So chemical applications are maybe some additional practices used for managing pest or weeds or pathogens. So when you say land preparation practices, tillage practices, selection of resistant varieties, then application of fertilizers, then fertilizers are sometimes chemicals, that is true. But if you apply organic fertilizer, organic amendments, they are, of course, non-chemical methods. But what you consider as chemical methods, pesticidal methods or all chemicals? If you think fertilizers as chemicals, yes, they are chemicals. Then fertilizer application, balanced fertilizer application is a cultural practice. Okay, then is that the only question we are having? Nothing you can ask from verbal? Then, okay. Thank you for your attention given for a longer day without a break. I hope you received some valuable information, clear explanations about some aspects of pest and disease management. Now, pay your attention to the weeds, weed biology and ecology, because as we have studied about the plants in many subjects, crop physiology, crop botany, I think by reading the weed biology and ecology section in the module, you can easily understand. So, because of that, I have taken more attention, I have paid more attention to the insects and the plant pathogens. Okay, so thank you very much for the attention given by listening to me passively or actively. Anyway, I am thanking you, and I will be thankful to you if you paid attention to the information, materials given in the module as well. Okay, have a good time.
Sir?
And yes,
Sir, we can use chemical methods and cultural practices together to manage disease management like IPNS method.
yes, of course. And chemical methods are a part of integrated pest management also. IPNS, integrated pest and nutrient system. If you say IPNS, when you say INS, integrated nutrient management system, you are applying nutrients considering the requirement of the crop and the medium. So both are equally valid for pest management also. Yes, you are correct. Cultural
Thank you.
practice. Yes, it is a good suggestion, good idea about remembering that. Yes, thank you for reminding that and clarifications. So, okay. Thank you for your cooperation, attention given. Hope to meet you maybe at the examination. If you would like to meet again for a short meeting, I am not having any hesitation. Send the request maybe through your representative, then we can have a short, maybe another meeting, maybe in an evening session. Again, I am not against to have any extra time if you would like to clarify anything about the materials we have discussed today. Maybe you are also tired, hungry, so maybe we can have an extra time as well, right? Okay. All the best, good studies, good health, and so you can have holidays next week and come for the examination on 6th. So next week's weekend, we don't have examination. Not on 31st. Right? Almost 3 o'clock. Have a rest and studies as well. I'll wind up the meeting now. Thank you. It was a pleasure with you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.